Okay, folks, here we go again. This time we're going to continue from where we left off last time, which was talking about Gregor Mendel and his genetics. And we're going to focus on Mendel's pea plants today. So he used to grow peas in his garden. And remember, the pea plants come in two varieties. There's green and yellow. We saw pictures of those in the last video. So the color of the plant is obviously a trait. And this trait is passed on to what we call the offspring, the pea plants that are baby pea plants, I guess you could call them, when we cross these plants. Okay? So, one of the things we have to do is keep track of what we call the generations. If we're going to start crossing pea plants and having baby pea plants, and then those baby pea plants, we can plant those and grow more pea plants and then breed those pea plants, we end up with a whole bunch of what we call generations. So when we're talking about the generations of a genetic cross, all right, so we're talking about what happens when you cross. I'll put the word cross here to remind you. We know what that means. So you take your first individual and you cross it with another individual, and you get what's called the parent generation. And so that's usually called the P generation, capital P for short. These are just a bunch of notations we have to get used to. Well, those parents then are going to have babies. So the next generation would be what we call the F1. F1. And the F comes from a really weird word called filial. Filial. Filial just means your children. Okay, so F1 would be the children of those parents. Now, of course, if we took some of those children pea plants and crossed them again, we could get the F2 generation and the F3 generation and so on as long as we wanted to. So if I draw this in sort of like a picture, it's like a family tree where you have, we'll call it a mom crossed with a dad. All right? Now, with plants, it's not really a mom and a dad because plants, as we see, we saw it in our last unit, they have basically very different sexual reproduction than we do. So they don't necessarily have a male and a female individual, but they have male and female parts. But it's the same concept, so we can just pretend. For humans, of course, though, you have a mom and a dad, and when you cross them, you might get, let's say you've got 3 children. So here's child 1 and child 2 and child 3. Okay? Now, obviously with people, children don't cross with each other. That's kind of a bad thing. But with pea plants, you could certainly take one of these pea plants, say this one, and cross it with that one. And if you did, you'd have another generation and all of the pea plants that were produced from that one. This is the parent generation. Here in the middle, we have the first filial or F1 generation. This would be called the F2 generation. And if you took this one and you mated them again, you could get the F3. So it's basically just successive mating of the offspring over and over and over. That's what the P, the F1, and the F2 simply means. So that's pretty straightforward. So you'll see that on our concept list, we have generations there. Now we understand what that means. The other thing on the concept list is what we're going to do today. We're going to do what's called a monohybrid cross. And what that simply means is that all we're going to do is keep track of one particular trait. Just one. And that trait today is going to be the color of the pea plants. The color of the peas. You can imagine if you were doing this in a complicated way to determine the outcome of something as complex as an entire individual and all their traits and all their behaviors, you'd have to do one heck of a huge database. But we're just going to do one very simple just to get the idea, and then we can see how that could get more and more complicated as we go. Okay, so the color of pea plants. Well, there's yellow and there's green. Those are the two colors that they come in. Mendel saw that. But there's something really interesting about that. When he breeded yellow and green plants over and over and over, he noticed something. He noticed that yellow plants, whenever they bred with green plants, produced way more yellow plants. There was something about yellow that seemed to be blocking out, hiding, or masking green. And so from that, we said that yellow is the dominant trait. It tends to occur more often than the green, and the green in this case is the recessive trait. Now remember that traits are caused by something called alleles. So each factor or gene, let's put it this way, each parent produces gametes by meiosis. Okay? And remember also that each parent possesses two forms of the gene. Or we could say two alleles of the gene. Now, we don't really know yet what the parent has. The parent could have two dominant alleles, it could have two recessive alleles, or it could have one of each. There's many ways that happens. But in meiosis, the two alleles are spread apart. So during meiosis, the two alleles are separated. You'll recall, separated. You will recall when we talked about meiosis that what happens is inside of the cell, in the first division of meiosis, the homologous pairs of chromosomes were pulled apart. And those were the two chromosomes that did the same job. Well, now we understand that each of those chromosomes would have a different allele, a different version of the gene on it. So when they're separated, the alleles are separated. And each parent is only going to give one allele. So each parent donates one allele to gametes. Remember what gametes are? Sperm and egg. So every sperm cell a man produces has one of the parent's alleles in it. And every egg cell that the female produces has one of the female's alleles in it. Okay, so we're going to draw a picture of that. We're going to have a male and a female. And for any particular trait, right, we said that the male has two of them, two alleles. And we don't know. So maybe this male has one dominant and one recessive. And maybe this female has one dominant and one recessive. Maybe she has two recessives. Maybe she has two dominants. When this female and male are going to produce sperm and eggs by meiosis, these will get separated. So every sperm will either have a D in it or an R. It'll either have, in other words, each of these is like one of the pairs of homologous chromosomes, just one of them. So they got separated, right? And over here, it'll be the same thing. The eggs, meiosis, will either be dominant eggs or recessive eggs. You can't have an egg that has both because in meiosis, they're pulled apart and separated. So the alleles are separated. And now when we cross them, we want to sort of see what will happen. Okay, so let's go back to Mendel's pea plants. We had said that we had yellow peas and green peas. And Mendel was able to identify that yellow was dominant. And he did this by using, remember we talked about true bred yellow plants. So you cross yellow peas, you get more yellow peas. You cross those ones, you get more yellow peas. And you keep crossing and you never see a green pea. So those are true breeding yellow. And what that means is that they'll only produce yellow, which means they carry none of the alleles for green. Likewise, he found out that green was recessive, and he found that out by also breeding true-bred green peas. Okay, now we need some symbols. We can't just keep calling it dominant, recessive, D and R, because it's different traits. So what we do is this. We take the dominant trait, which in this case is yellow, right there, and we use the first letter of yellow, and we call it capital Y, to represent the dominant allele, the form of the gene that produces yellow. Now, the green is the recessive, but we're not going to use a G. We're going to use the same letter because we want to understand that these belong to the same trait. They both belong to the same trait of color. So for the recessive, we're going to use a small y. So we have what are called alleles, dominant, recessive, and we're using a small or a large letter to represent them. And we always go with the letter, the capital letter of the dominant gene. And then the other one is always the small letter. Okay, so let's do an example. Let's take a true-bred yellow plant. True-bred yellow. What that means is none of the peas in this group have any alleles for green. That means that they will have, the parents will all be capital Y, capital Y. All right? Now we have a term for this. We call it homozygous. Whoops, I'm sorry. I better erase that. That's not an S. It's supposed to be a Z. There we go. Homozygous dominant. The word homozygous is a big fancy word that just means homo means the same, two of the same alleles. So homozygous means having two of the same alleles. Okay. True-bred green would have only green alleles. So true-bred green peas that never made yellow would both have two alleles like this. And that's called homozygous recessive. Two of the same, but it's the recessive ones. Let's see what happens if we were to cross two of these plants. So the way we cross is we do what's called a Punnett Square. This is named after a fellow by the name of Punnett, who came up with the idea of doing a square. It's really just a chart. It's nothing really fancy. And it looks like this. You draw like you're going to play tic-tac-toe. Okay. On the top, you write one of the parents' alleles. So remember, if I could go back there, we'll see that the true-bred yellow, there it is, two capital Ys. So when it goes through meiosis, it will make egg cells or sperm cells that only have capital Ys in them. So I put the capital Ys here across the top. The other one is homozygous recessive. It only makes sperm or eggs that carry the green version. I'll put those ones down here. And now when we cross them, we simply work across. So we look across this way, and we look down, and we match them up. And we always write the capital letter first, because that's just the way it's done. So what happens is when a sperm and an egg like this were to meet, they would produce an offspring that had one of each allele. And if I go across to the far one, you'd see it's the same thing. And then again, down below, same thing. And lastly, same thing. So we're just going across and down and matching up in each column. So these are the results. The two things that we wrote here and here were the parents. These are the F1 generation. The possible babies you could have. Now remember what I said before. This doesn't tell us exactly what babies you will have. It tells us the ratios. If you look at our options, the ratio is we're going to have 100% of all babies formed will all have to be this. There's no way to get 2 capital Y's or 2 small y's when we cross these parents, because all of the sperms have capital Y's and all of the eggs have little y's, or vice versa. It doesn't really matter if these are the sperms and the eggs on one side or the other. Okay. What do we call this? Well, it's not homozygous because it has 2 different alleles. So we call it heterozygous. And that means it's neither dominant nor recessive. We don't have to add the word dominant or recessive because if they're 2 different ones, that's just the way they are. But how would we be able to tell the difference between them? So let's look at the options. We had this one, and we had this one, and then we had this one. This one, I'll just use a short form. Homozygous, well, maybe I'll write it. Homozygous dominant. Homozygous recessive. And these are all just big words that are describing which of the 2 alleles we have, which of the 2 versions of the gene do we have, right? And this one is heterozygous. And again, it has neither. What would they look like? Well, obviously, the first one has only alleles for yellow, so it's going to look yellow. The other one has only alleles for green, so it's going to look green. Now, you might think that this one is going to look sort of yellowish green, but that's not the case, at least not in all genes. There are some genes that do. They blend together and it would produce a yellowy green, but other genes don't. Because this is a dominant and recessive allele, the dominant allele masks the recessive. So the dominant yellow gene is going to make this pea plant look yellow, exactly the same as this yellow. So by looking at it, you would never know which one it was. That's what dominant means. If a trait is dominant, like this one, that means that it will mask. So in other words, this one is like it's not even there. But it's important to keep track of, because if we have more babies out of these babies, those alleles will still be thrown around into the mix. So we have more words. We have words to describe what the actual letters or alleles are, and we have words to describe what the actual plants look like. Okay, so the genotype describes the actual alleles. Actual alleles. So our genotypes were homozygous dominant, homozygous recessive, and heterozygous. Those are the 3 possible genotypes. But we also have a word called phenotype, spelled with a PH, which describes the outward appearance. Outward appearance. So what we would have yellow or green. That's what we would see with our eyes. Now we know that the green one is also this genotype. But the yellow is confusing because these 2 would look identical, and there's no way to tell them apart unless you could investigate the actual genotype. So I wonder how Gregor Mendel told them apart. I wonder what he did. Well, guess what? He did more crosses. If you go back to our cross, where we crossed a true bred, right there, a true bred yellow with a true bred green, we got 100% heterozygous, but of course they would all look yellow. Let's see what happens if we did a different cross. Let's see what happens if we were to cross, and we'll just do a cross like this. Okay, so we had sperm, and the male was homozygous yellow again. But this time, we had another yellow plant. But its genotype was this. So we sometimes write it on the side here, YY cross YY, like that. So we want to know what's going on here. Well, if I just jump down a bit and just kind of show you what's happening, if you look at this individual, when they go through meiosis, they're going to separate their alleles in the first meiotic division, and they're going to produce sperms. Well, this one can only produce capital Y sperm or eggs, but this one can produce both Y sperms or Y eggs, right? So it's carrying this little Y, remember, he's not seen, he's masked out. So it's like we don't know he's there because he's recessive. This one dominates him. But when you make the alleles, when you make your sperm and egg cells, he gets to go all by himself and he's no longer dominated. So he's free to join in and see where he goes and what happens. So let's see what happens. If we cross this one, if we cross the first one this way and down, we get 2 capital Ys. The next one, 2 capital Ys. The next one, and the last one. What this is we get 50%, or half of the 4. There's 4 there, so 2 out of 4. 50% are this, and the other 50% are this. So that means that if you took these plants and had babies, then it doesn't mean you'll get exactly 4 babies and you'll get exactly 2 and 2. What it means is, if they had a whole bunch of babies, you would end up seeing in the population of their babies 50% of each overall. Okay? It works better, of course, when you have hundreds and hundreds of babies. If this was just a human only having 1 or 2 children, these Punnett squares will not be able to tell you exactly what the child will have, but only what its chances are. Right? So that's what you have to remember. These are percentages. They're like the chance of being something. They don't tell us exactly what will happen. So that's different. These genotypes here are different from these genotypes way back here in the first cross. This one was 100% heterozygous. This one is 50-50. But notice something. I'll go back here. What color are these all? What's the phenotype? Because they carry a yellow dominant gene, all of these are yellow, all of them. And then down here, if we look at this one, we kind of have the same situation. The top 2 obviously are yellow, but the bottom 2 also carry a dominant yellow gene, a dominant allele. So these are all yellow also. All yellow. Now, what's different is, in the first one, we had all 100% the same genotype, and 100% the same color, the same phenotype. But in this cross now, we've got 100% of the phenotype, but we have a 50-50 genotype. So you see how it's going to be different. The actual alleles look different inside, but they all make the plant yellow. Okay. Let's do one more cross and see what happens. Let's take a situation where we took some of those babies. Some of those babies were this. They were heterozygous. And then let's take the other babies who were also heterozygous. So if we go back to our peas, we took one of these and one of those, and we crossed them to see what kind of seeds they would produce. Let's do the cross. Capital Y, 2 dominant alleles. This one would produce 1 of each. The bottom one is 1 of each, but the last one produces 2 small Ys, 2 recessive allele combinations. When that sperm and that egg join, they make a recessive allele combination. Now let's look at the percentages. We may have to move a bit. In order to get this, well, you can see in there only 1 out of the 4 is that combination. That's 25%, 1/4. Then if you look here and here, there are 2 out of the 4 that give us the heterozygous situation. 2 out of 4, or 1/2, that's 50%. And then looking back, there's only 1 combination out of 4 that gives us the homozygous, they're both the same, homozygous recessive combination. That's 25%. And of course, that adds up to 100, right? But look at the phenotypes. So those are the genotypes, right? Those are the different genotypes and their percentages. They're not all the same, and they're not 50-50. This time they have a different combination, but they're all there. Now let's look at the phenotypes. The top one here, I'm just going to erase that 100% for a second, and we're just going to look at those colors. The top one is yellow. That's its phenotype, because look, it carries both yellow alleles dominant. But the second one is also yellow because it carries one of the dominant yellow genes that hides the recessive one, and it makes the plant yellow. But the last one is not yellow at all. It's green. This is the first time we've been able to see a pea turn out green. So we can tell, if we did this cross ahead of time, we can predict what would happen if we cross these 2 plants together. I could predict that I would get the following percentages of genotypes: 25 homozygous dominant, 50% heterozygous, and 25% homozygous recessive. But I could also predict the phenotype ratios. The phenotype ratios are, if you look at it, yellow is 50 and 25. So it's 75% yellow. So what we call the phenotype, that's an N, the phenotype ratios are yellow. You add up the percentages that we had up there, 50 and 25 for 75%, and 25% for the green. So you see, these are not quite the same as the genotypes because of that dominant and recessive combination that also looks yellow. Basically, what it means is you're going to get a lot more yellow than you are green. It's very hard to get green because there's only one combination of alleles that will get you a green pea. Okay, so what I'm going to do now is I'm going to show you how we could do some sort of examples together. And I'll write them down, and then you can pause the video and you can do it, and then we'll take it up. So let's do a cross. Let's cross a homozygous yellow crossed with a homozygous green pea. And what we want are A, the genotype ratios. What can we expect to happen? And B, the phenotype ratios. Okay, so I'm going to just set you up and then you can pause. So first of all, what do we have? Homozygous yellow. We have to know what that means. It means capital Y, capital Y. And homozygous green means little y, little y. So you have to know that. And then you make your chart, your Punnett Square, like this. And you put the 2 alleles from the first one, the 2 capital Ys. And it doesn't matter whether you put them on the top or the side, as long as you put them on the same spot. And the other one goes on the other side. Okay, now what I want you to do is make the crosses and tell me what the genotype ratios are. And then I want you to think about what color they would be and tell me what the phenotype ratios are. So pause the video, try it on your own, and then come back when you're ready. And you're all set. For me, it was a half a second. For you, it was a couple of minutes. So what you should have done, let me just try a different color here. Let's try this nice blue color. You should have written capital Y. Oh, that's a terrible color. Let's go back to pen. Here, this is what I want. Let's go back to here. This is better. Capital Y, small y. Capital Y, small y. And remember, all we're doing is reading across this way and down this way. And wherever the columns match, right, you put the right ones in. Capital Y, small y. Capital Y, small y. They all look the same. So the genotype ratios, the answer to part A, is you're going to get this 100%. And the phenotype ratios would be what color they are. Since they all have a dominant yellow allele, everyone is yellow. So yellow, 100%. Okay, that's the answer to that one. Let's try one more. Go back to black. Let's do, this one will be a heterozygous yellow. And we're going to cross that with a homozygous green, P. And we want to figure out what the baby's chances are of being yellow or green and whatnot. So again, A, figure out the genotype ratio, and B, figure out the phenotype ratio. You know what that spells. It spells phenotype ratio. By the way, I found out on the internet that a caret works just like a finger on an iPad. And so believe it or not, I'm writing with a sharpened caret because it feels more like holding a pencil in your hand. My writing was actually worse with my finger, if you can imagine that. Hopefully, as I get better with the caret, I'll get neater and neater and neater. Yep, a caret. Told you I was crazy. Okay, part A. Well, the first step. Go back to the question. What does it mean? Well, homozygous yellow means, heterozygous yellow means one of each. It's yellow because of the dominant allele, but it has two different alleles. And homozygous green means it has to have two of the same, and they have to be the recessive allele for green. Because yellow is the dominant trait. Why is yellow the dominant trait, you might ask? That's got to do with the chemistry of the genes and the way they interact with each other. So Mendel couldn't really answer that question. All he knew was this is how it worked, and he didn't have an explanation as to how one gene could mask the other. We know now, and it has a lot to do with the way genes are expressed and turned off and on by the biochemistry inside cells. But don't worry about that just yet. So now we have to make a Punnett square using these two. We're going to cross those two. So we'll do that, and we'll cross. Okay, so I'll just go back to double check. We have a heterozygous and a homozygous recessive. So here's the heterozygous, and here's the homozygous recessive. Let's cross them. Big Y, little y, two little y's. Big Y, little y, two little y's. Okay, what do we got? Well, you'll notice that we have in here, out of the four, there are two out of the four that are heterozygous dominant. That's 2 out of 4, right? Out of the 4 boxes we made, which is 50%. You'll also notice that there are 2 out of the 4 that are homozygous recessive. That's also 50%. So those are the genotype ratios. We'll get 50% that are this combination and 50% of that. You get no other combinations. No 2 capital Y's. You don't get that one. They don't exist. You cannot get a homozygous dominant baby out of these 2 combinations. These parents could never make one. Okay, now the phenotype ratios. Well, if you look at the red one, the heterozygous one, because of that dominant gene, they're going to be yellow, that dominant allele right there. But the other ones are going to be green. So now we have a phenotypic ratio of 50% yellow and 50% green. Sometimes the phenotypic ratios match the genotypic ratios. The phenotype ratios down here are 50-50, and so are the ratios up here for the genotypes. But sometimes they don't, as in our other example. And that's how you do a monohybrid cross. And let me just have a quick look at our list. We've talked about genotype, phenotype, Punnett Square, generations, and monohybrid cross. But we haven't specifically said in detail, which I will because it's kind of important. Although we've used it, we just haven't written it down, is the law of segregation. So Gregor Mendel, remember, he didn't understand segregation. He didn't understand what was going on deep inside. He didn't know the genetics. It wasn't discovered yet. So he just knew that somehow there were these two factors or genes, and somehow they had to be passed on from the mother and the father, and that they had to be separated or dissociated. So segregation means to be separated. And this refers to the fact that when you have an individual who has, let's say, this pea plant, then when they make gametes, when they make sperm and egg, in the first division of meiosis, those two homologous chromosomes will be pulled apart. And one sperm will get one and one sperm will get the other. That's the segregation, right? And we understand this better with our knowledge of genetics that he didn't have. Poor Mendel had to just figure this out. So the law of segregation in words simply says that traits, I'll write it out here for you. Here's a good example. Traits are determined by pairs of alleles. And we can write in brackets, because now we know what that is. It's homologous chromosomes. That's why there's two pairs of alleles. Each homologous chromosome has one of the alleles. They both do the same job. They both determine the color, but they do it in a slightly different way. So traits are determined by pairs of alleles that segregate. And we also know now how they do it. Segregate. Mendel didn't know this, but we knew about the process of meiosis, which is a kind of cell division that pulls apart the homologous pairs of chromosomes. Right? So in brackets are our current understanding that segregate through the process of meiosis so that each gamete, sperm or egg, made by the parent receives only one allele, only one form of the gene. Okay, so now you're ready to do some monohybrid crosses and to figure out some genotypic ratios and phenotypic ratios. And so in class 2, I'm going to soon be posting a little question sheet where you can do things just like we did in our example up here, where you had to figure all this out. And you're going to try to do that, and you're going to just practice that a few times, fill in the sheet. Perhaps what I'll do is I'll find a way, I'll set up the Dropbox so you can actually hand in your results. You can hand them in any form that works for you. If you want to take a picture of your work, which might be the easiest because you're just going to do it on paper because it's charts and graphs and things. So just write it all out, take a picture, and email it to me if you want, or you can put it in the Dropbox on LMS, whatever works. And if you have any trouble getting me the assignments, just let me know and we'll figure it out. That's all for today.